right, so today, we're still on chapter 2, all this biochem stuff. I just wanted to go back. I want to make sure that I highlight some of the important parts of functional groups. I also tagged on a little section in the new slides I just posted yesterday morning on pH, because I had a lot of questions about that. So I want to make sure we've got that down, and then we'll go into macromolecules. Okay, so the reason why I want to make sure we understand functional groups is because they make up the big biological molecules that we're going to talk about. Okay, so when I say the 4 classes of biological molecules, that's proteins, carbohydrates, nucleic acids, and lipids. So that's what you're comprised of. So here we are talking about atoms, right, and elements, and we're going to put all those together, and we made compounds, and we made molecules. You're going to the next level now, and you're going to build these proteins, and you're going to build carbohydrates. And then you're going to see where do those fit in the body. Okay, so this is important stuff. These functional groups are going to tell you the properties, okay, that they have that then lend themselves to a lipid or to a protein for function. So that's why it's important. So I listed them here, but let's go through them again. So hydroxyl. First thing to tell you is you're seeing it written like that. Oh, not with this marker again. That's the same thing as that. For some reason, when students see the line, they get all confused. Or if the line is gone, they're confused. Okay, that's the bond, right? Remember, that's the covalent bond. That's all that is. So don't let that freak you out if you don't see it. Okay, so if you have a hydroxyl group, which is just oxygen to hydrogen, and I know you don't have this all memorized, but hydrogen had a valence of 1 and oxygen was 2. So hydrogen can form a covalent bond with one other atom, and oxygen does it with 2. So that's what you're seeing right there is, here's this bond, there's this bond to probably carbon. Okay, so where are you going to see those? Hydroxyl groups are important for solubility. Your body is mostly water, and we said that we're going to transport all these things in your body through bloodstream. Your cells are water on the outside, water on the inside. So you need things to dissolve. Okay, if you eat something and you've got preservatives and toxic chemicals in there and some kind of dye, your body's trying to get rid of it, and one of the things your liver is going to do is take it, put these hydroxyl groups on it, okay, it's going to detox it, then kick it back out into the bloodstream where it's going to be soluble and your kidneys will excrete it in your urine. Okay, so that's hydroxyl groups. They make things soluble. Typically, if we see a molecule with a bunch of hydroxyl groups, we call it an alcohol, like methanol, propanol, and so all those fun things, methanol. Okay. A carbonyl group, there are 2 types. These are important today because we're going to be talking about them in carbohydrates. Your carbs are sugars. You have simple sugars, you have complex sugars. We'll talk about all of that, but what defines it is we see these carbonyl groups. So that's kind of its property. If you have a carbonyl group, you're a sugar, you're a carbohydrate. It's a carbon that's double bonded to oxygen. So there's oxygen has completed its valence. Hydrogen is attached to carbon, it's completed its valence. And carbon's bonding is 4. So there's its 1, 2, 3. This is probably another carbon over here. So there's 2 types of carbonyl groups depending on where they're found in the molecule. This one, we'll say it's on the terminal end, and you'll see pictures of this today, I think maybe. If it's on the end of your sugar, it's called an aldehyde. Or once it's in an actual sugar molecule, you might have heard of it as an aldose. If it's in the middle, so if you have a carbon and then the carbonyl group and then another carbon, it's called a ketone carbonyl group. The whole sugar is called ketose. So you guys have heard of ketones and ketose? That's what that is. Carboxyl group, I told you, was the hydroxyl. There it is, shorthand, right, without the line, attached to a carbonyl. So all we did was put them together, carbonyl plus hydroxyl. They had a baby, carboxyl. This one makes something so that it is polar. Okay, so it gives it the properties of being more polar. It's an acid. Okay, so an acid, if you remember what that means, it can donate a hydrogen into solution and increase the hydrogen concentration of that solution. So if you drop this in the bloodstream, it could lose this hydrogen. This would have a negative charge here. Hydrogen would be H+. Why does oxygen get a negative charge? Because why? Gained an electron. Oxygen's negative because it gained and then it took hydrogen's negative and kicked off the proton. Okay, so that would be your carboxyl. So these act like acids. The amino groups act like a base. Okay, and it looks weird. And I know it's not going to make a lot of sense. I can explain the chemistry to you, but if you take another hydrogen on, which is what it does, it becomes NH3. Okay, so it takes hydrogens out of solution. That's what a base does. So it reduces the hydrogen ion concentration. So you have aminos, that's your base. You have carboxyls, they act as an acid. Esters, this is a carbonyl attached to an oxygen and then another group. You're seeing this R. You're like, what the heck is this R? That just means it's a general side group that you're going to see, or another, it's usually a carbon. Okay? But it's just shorthand for generic. Those are in your lipids, so we're going to see those in, there's different types of lipids. A phospholipid, there's triglyceride. Sulfhydryl groups are in proteins. It's a sulfur with a hydrogen attached to a carbon. Okay? And those make your hair curly. So if you have curly hair, you have disulfide bridges. You have two of these groups attached to each other. So if you want to straighten your hair, you're going to break that bond with a really hot iron, and then you're going to walk outside in Texas where it's humid and put water back in your hair, and it's going to go whoop and curl right back up. Okay. Phosphate groups are energy molecules. If you tag a phosphate onto another molecule, these have a ton of negative charges, what they conveniently did not show you here. Normally they're ionized in the body. You don't have a hydrogen here or here, you have negative charges. So when you tag these negative huge phosphate groups onto a protein, it makes it highly reactive. Okay? And we see these on your DNA, your RNA, and ATP. Methyl group I wanted to put in here. These are completely non-polar, no charges, because carbon and hydrogen have equal electronegativities, if you remember that from Tuesday. They share the electrons equally, so no charge. These you often find on your DNA. They sit on our genes and they silence them. Okay, so the idea is you don't want all of your genes being transcribed into proteins all the time, right? So this is the way that we kind of decide which genes you're going to turn on or off. Okay, so that's a little intro for that. Let's just revisit real fast for you people that asked about pH. I'm not going to go into a ton of it, but I just put the slides in there for you so you can practice. This is what pH is, the negative log of the hydrogen ion concentration. And I put a little note. Remember, pH is going to be a whole positive number, like 2, 3, 4, 6. It's not going to be 10 to the negative 7. Concentrations are in our class are always going to be 10 to the negative something molar, okay, or written out like this. So in the problem, if they give you the hydrogen ion concentration, and they said, okay, you have hydrogen ion concentration of 10 to the negative 4 molar. What is the pH? It's just the exponent, 4. Okay, it's that easy. Remember, it's the negative log, so that's how you get rid of that negative. If you're given the hydroxide concentration, and they tell you it's 10 to the negative 4. That's not the pH, that's the pOH if you said 4, right? So how do you figure out the pH? Remember, these exponents for hydrogen ion and hydroxide have to equal 14. So what is 4 from 14? It's 10. So hydrogen ion concentration would be 10 to the negative 10, which means the exponent would be 10. It would be 10. What if you're given the pH and they ask you for the hydrogen ion concentration? So they tell you it's 4. It's just 10 to the negative 4. Okay. What if they give you the OH, hydroxide ion, and they said, what's the pH? And they said the pH is 10. How did we figure that out? Okay, well, if the pH is 10, hydrogen ion is 10 to the negative 10. Well, remember the rule of 14. 14 minus 10, this would be 10 to the 4th. So that's how you work that. You just got to be careful and say, what is the problem giving me and what is it asking? Be careful. Hydroxide ion is not the same thing as the hydroxyl group. This is an ion, which means it's floating out in solution with an extra electron. It's negative. A hydroxyl group is covalently attached to a carbon. It doesn't have that charge. Okay. All right. Time for biomolecules. Do you guys have any questions on any of that stuff? I'm going to work on a nice review for you so you know exactly how to sort of work through the material, because I know you don't have any Practice tests. I don't have any practice tests. I asked Dr. Jackson for stuff, but I didn't receive it. And she's pregnant and just had a baby, so I'm not bothering her. Okay, so we're just going to make it work. Okay, so here's our 4 main categories of our biological molecules. This you absolutely have to know. If you walk into the test, you're like, I don't know what the 4 classes are. You're in big trouble if you can't just list them off the top of your head. Okay, so carbs, lipids, proteins, nucleic acid. Okay. They're molecules. So what does that tell you about the bond between all of the atoms in these structures we're going to look at? Covalent. They're strong covalent bonds. You got it. Okay. And notice, again, with this damn textbook, they're like, this is the section of macromolecules. No. Why is that wrong? Because lipids are not a macromolecule. Technically, the word macromolecule means a big molecule, but that is not exactly what it means in biology. It means that you have a repeating unit over and over and over to make a big polymer. Lipids are not polymers, so they are not macromolecules. So they're biological molecules. Before we get into these classes, you're going to learn about them. What are they made out of? Structures, function. So we learn the structure, and then we learn the function. We have to learn how we make them. Okay, because I was saying most of these are going to be these repeating units, okay, like amino acid over and over and over. How are we sticking them together? It's called a dehydration reaction. Okay, and sometimes in chemistry they call it a condensation reaction. It's the same thing. Okay, so this is how we stick 2 things together, 2 subunits. We're going to call these monomers, but in the lipids you're going to see this where they have a dehydration reaction and build something bigger. So we're forming a covalent bond. Why is it called a dehydration reaction? When you're dehydrated, you have lost water, and that's what's physically going to happen here. And I just put a little cartoony thing in here. You're going to see a hydroxyl group from one of these structures and a hydrogen from another structure are going to combine and form a water and leave. They're gone from this molecule. And then a bond forms where these would have been. So where this hydrogen would have been and here come together to form a bond. Okay, so you don't have to know about all the specifics of how the bond is formed. Just understand when we're building something together, that's a dehydration reaction. We're going to lose a water. And there's a specific dehydration reaction for every class. Okay, so we're going to make sure you know those. This is my shorthand for a reaction. Probably not going to get through all this today, but maybe. So as we go through, I'm going to list them for you. The opposite of that, so think of a dehydration reaction. You are building a bone, you're building a muscle, you're building a cell wall in a bacteria. Those are all dehydration reactions. Your body is constantly breaking down, so you have to repair it and build it back up. If you're growing, you've got to grow, right? You've got to build these things. On the opposite side of that, we also have to break things down. If you were lucky enough to get some breakfast in your stomach this morning, you didn't just eat a bagel and have the whole bite of bagel go across your stomach wall into your bloodstream, right? That doesn't happen. You have to pull it apart into all the little tiny pieces before it can go into the bloodstream and into your intestines and then dissolve. So these are called hydrolytic reactions. Okay, so dehydration, put them together. Hydrolytic, break them apart. Lytic usually means to lyse, which is break apart. Hydro, again, is water. So in this case, we're actually bringing water in, and then we're pulling it apart. So that's why it's called hydrolytic, okay, or hydrolysis. They're the same word. So you're going to bring water in and pull it apart. So a hydroxyl group goes one way, a hydrogen the other on either side of this oxygen. So now you have 2 separate molecules. Okay. Okay, so this is what I was telling you earlier about macromolecules. They're assembled from a repeating unit. So get comfortable with this term, monomer. That's your repeating unit. So if we were in a protein, it would be an amino acid. If we're in DNA, it's going to be a nucleotide. It's the unit that attaches over and over and over and over. Okay, so notice it's only carbs, proteins, nucleic acids. Lipids are not macromolecules. They are very large, but they don't repeat over and over and over. That's not how it works. Okay, so these monomers are going to link up until they form a polymer. Poly is many, so many monomers. And the example that I gave you was pop beads. Okay, if each bead was a monomer, you're going to pop them together, which would be your, or stick them, I guess you're not popping them together, is your dehydration reaction until you form the polymer. Okay, so that's essentially what you're doing, but you're going to do that with these materials to make carbohydrates and proteins and nucleic acids. So you're going to need to know the monomer unit, and, well, you need to know the bond for all of them, but you need to know what is the monomer unit for each of these, and then what is the polymer? What do we call it? So we're going to start with carbohydrates. Okay, carbohydrates are sugars. Okay, so when someone is drinking a beer and they tell you you're on a diet, you're like, you're drinking straight sugar. Okay, or if they're on a diet and they're doing no fat, but then they're eating a bunch of carbs, you're eating straight sugar. Okay, so it doesn't matter what kind of diet you do. If you eliminate one of these groups, that is bad for your body, and it's never going to work. Nature is all about balance, so you just need to balance things out. So you shouldn't get rid of any group. There are two kinds of carbohydrates depending on their structures. Simple carbohydrates are the ones that you see on your food label as sugars. So if you're looking on something, you're like, carbohydrates, and then it says sugars under the total carbs, it's telling you the type of carbohydrates that break down really fast and absorb right into your bloodstream because they're so small. Okay, so your monomer unit for your carbs is a monosaccharide. Okay, saccharide means sugar. So this is a monosaccharide. It's a simple sugar. What defines it as that little unit is that it has 3 to 7 carbons in it. And it's going to have a carbonyl group. So anywhere from 3 to 7. Beyond that, we don't have any monosaccharide structures. We don't get bigger than 7 carbons. And an example of a monosaccharide would be glucose. Disaccharides was if you took 2 monosaccharides and did a dehydration reaction, stuck them together. So glucose that you find in your body for fuel, and fructose, which you would find in fruit that tastes sweet, if you put those together, each of those are monosaccharides. If I attached them, they would form sucrose, which is that white table sugar that you're used to putting in your coffee. That's a disaccharide. So monosaccharides and disaccharides are all still small, and they taste sweet on your tongue. Okay, so that's how we classify those. Those are simple, tiny sugar carbohydrates. Once they start getting bigger, which is beyond the 2 monosaccharides, okay, normally you don't see 3 or 4. It's either 1, it's 2, or we've got hundreds. Okay, they just link and link and link. So I'm going to take glucose, 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 over. That is called a polysaccharide. Okay, so that's going to be your polymer. So monosaccharides are your monomer, polysaccharides are your polymer for carbohydrates. What's the bond that's going to connect these monosaccharides to each other? It's called a glycosidic linkage. And you should have learned all of this in your bio class. So here's some structural pictures you can see. Glucose. And there's 2 different ways that we draw this. This is what it actually looks like in your body. This is sometimes how we just draw it on textbooks so we can just look at it. Here is the carbonyl group that defines it as a sugar. Okay, and it's called an aldehyde because it's on the end of this carbon. If it was on the end of that carbon, it would still be an aldehyde. But if it was attached to any of these carbons in the center of the chain, we would call it a ketone. Like up here, this is on the 2nd carbon, so it's a ketone. You guys see that? Okay. So glucose. And then remember what I was telling you with the ring structure? They won't show you the carbons or the hydrogens, but you'll see an oxygen. That's what it would look like. So here's a monosaccharide, here's a disaccharide, and then here is what polysaccharides would look like. So do you see the monomer unit now? It's a little bit easy to see the monosaccharide attached over to make a big polysaccharide. You are not going to have to identify the structures. Okay, so you don't have to worry about that. I do want you to know that glucose is the very most important simple monosaccharide or simple sugar that you can have, okay, for all life on the planet, because that is fuel for our body. I shouldn't say all life, those of us that use glucose. Okay, so that's really important. Glucose is like money. If you guys had a lot of money, but I said, here's $100, would you be like, no, I'm good? No, you'd be like, sweet, $100. That's what your body does to glucose. It's like, I've got tons of glucose. I've got it all stored up. And then you eat a Snickers. Your body's like, awesome, glucose. I'm going to put this in the bank. And it saves it. But if it's like, oh, I've got a ton of glucose already saved up in my reserves, I'm going to convert it to fat. I'm going to save it long term. Okay? That's what your body does. That's why if you see glucose in your urine, not you're going to see it, but doctors used to taste the urine to see if it was sweet. Yeah, be thankful you don't have to do that anymore. You just stick the little stick in there now. But you should never see glucose in your urine because your body never excretes it. It loves glucose. So carbohydrates that are your small ones, the mono and the disaccharides, usually have the suffix OSE. So a hexose or a pentose, and you're hearing those prefix based on how many carbons are in the structure total. Hex is 6, pent is 5, so this is what it might look like for a 6-ring carbon or a 5-ring carbon. Okay? And notice, we'll say a 5-ring carbon, but notice the 5th one is sitting up here. This one is important. You can put a little star next to ribose. Ribose is important because he, little guy, is part of DNA and RNA. So when we get down here to nucleic acids, you're actually going to use a ribose in there. Okay? And when you get to how DNA is transcribed or replicated, you have to understand the whole 5' 3'. Like, what is that talking about? It's talking about this 5' carbon and this 3' carbon. So a little preview of what's coming. Okay, here's a picture showing you a glycosidic linkage. Here's galactose to glucose. Okay, and now you have lactose. So this is your disaccharide. You don't have to memorize these. I'm not going to ask you on the test, what is lactose made out of? So don't worry about that. Like I said, I'll make you a nice little review. Okay, so let's talk about our polysaccharides. Those were your simple sweet sugars, your tiny monomer units. Polysaccharides are your complex carbs. So when you see total carbs and you see sugars and then you see the actual carbohydrates, that's complex carbs. They're in a different class on your dietary labels because they're processed differently in the body. If you eat a Snickers bar, your body's going to chop it up really fast and put that glucose right into your bloodstream, and your brain is going to feel happy because that is fuel for your brain. Your brain can't store glucose, so if you don't eat, you're going to be dumb. Feed your brain, right? Okay, so get it some glucose in there. The polysaccharides, hey, these are big, and the body slowly breaks these down. These are actually better for your body, right? You don't want a big spike of glucose in your bloodstream. So it takes longer to break these down, that word hydrolyze. There's so many more bonds to break. Okay, so you're going to break a bond, then break another bond, then take bonds. Remember, that's energy to break those bonds. So what's the function? So what was the function of our simple carbs? Energy. Quick energy, okay, very fast, quick energy. Our polysaccharides have two functions. One is still energy. Okay? We can make glucose into a polysaccharide called glycogen and still use it for energy, for quick energy. The other one is for structure. A lot of animals don't have skeletons, so they can use complex carbohydrates to make an outer shell that is strong enough to protect them. So they make these exoskeletons. So let's look at the structures first. So cellulose is a really important organic compound. It's one of the most abundant organic compounds on the earth. And the monomer unit, and you should definitely know this, is glucose. So that means cellulose is made completely of glucose. When you're looking at cellulose, it's just thousands, millions, billions of glucose over and over and over through these glycosidic linkages. It's attached in such a way that we do not have enzymes to break down cellulose. So cellulose is things, like I was telling you, the strings in your celery. You can chew this and chew this and chew this, and it's still going to come out corn. The outer wall of corn. Yes, I have kids, so I can show you nice pictures of what it looks like when that comes out. It looks just like corn, right? It doesn't break down. We don't have the enzymes to break it down. In our bodies, it's a lot of plant material. Wood is cellulose. When you're burning wood, you're burning cellulose. So in our bodies, we don't use that as an energy source because we can't use it. We can't break it down. We can't get any energy out of it. We use it for fiber. Okay, so for us, when we eat something like that with plants that have a lot of cellulose, it's going to go through your intestines and gently abrade the walls, when as soon as stimulating those cells to produce mucus, and everything comes out. Okay, so that's healthy. If you're not going once a day, there's a problem. Okay, what about all the other organisms that actually use this for energy? Bacteria, there are fungi, protozoa. What if you're a cow or you're a horse and you're eating grass or eating hay for your food source? How are you getting any energy? They can't. Even a termite who eats wood, which is straight up cellulose, cannot break it down. So what they have are these bacteria that live in their guts that break it down. So there's bacteria that can actually take the energy out of cellulose. So the bacteria are in there, and they're breaking down the cellulose and releasing the energy for that organism. And then the bacteria are living off of whatever kinds of gases, like something like methane, for example, in the gut of those animals. So everybody's happy. It's a nice little symbiotic, gross relationship. Okay, chitin is your second structural carbohydrate. This one is a polymer of glucosamine. This one's a little bit different. It has glucose in it, but it's a little bit structured a little bit differently. So if you're a fungus, and here's a really cool photo of a ton of fungi, it's in the cell wall. If you're an insect, okay, it's the exoskeleton. We see these all over campus. If you're an arthropod, like a spider or a crab or a lobster, the outer shell, that's chitin. Okay, and if you see this around your house and you think, oh, thank God that spider's dead, probably not. If there's no little body attached, that means there's just a bigger spider crawling around your house somewhere because it just took its shell off and it's like, I'm bigger now. Okay. A cool part about chitin, cellulose, we said, doesn't break down in the body. Okay? Chitin will break down in our bodies eventually, but it takes a long time. So what some really brilliant people decided is, hey, we're going to take this material and make it into a surgical thread. So when we have to stitch somebody up internally, we don't want to open you back up and pull your stitches out. We don't want to use plastic or something that your white blood cells might attack, or you might form a biofilm on. So we're going to use chitin because it's this natural product that eventually is going to break down, but it's going to give your body time to actually fuse back together and heal. So it's pretty cool. This one we talk about because you're using it in lab. Agar. Okay, agar is a cell wall component from red seaweed, this little seaweed right here. You guys are going to see it on all your micro plates. Are you guys doing plates yet? Are you doing anything fun? Lab hasn't started? You're going to love micro lab, except for when you have to come in on the weekend. But to, work your bacteria. You're like, what? It'll be fine. Pay your lab mate. Okay. But this is what it looks like. You can melt it, then you pour your plates. Okay? And the story behind this, there's a lot of stories that we'll kind of talk about, but a lot of women are not mentioned in history because the scientist was their husband, but the idea came from their wife. Okay, so it was actually a wife that told her husband, hey, you know what you should try is agar for this one thing that he was doing, and now we use it for everything. So way to go for that lady. Another structural carbohydrate is peptidoglycan. You guys are going to hear about it all the time because you're in microbiology, and this makes up bacterial cell walls. So remember, a bacteria is a live cell, so it has a plasma membrane just like we do, but then it also has this awesome cell wall on the outside that gives it structure and integrity and protects it. So they have this peptidoglycan. We have not learned about proteins yet, but you've learned sugars. Glycose, saccharin, that's all sugar. So glycan is telling you that's the sugar part. Peptid is short for peptide, which is protein. It's a little tiny piece of amino acids. So this is actually a big bunch of sugars all stuck together with a little bit of protein stuck on it. So it's a sugar-protein molecule. Right? And this is just showing you, you're going to hear about NAM and NAG. I'm not going to ask you this stuff, but that's the sugar part. And then these are the little amino acids attached to it. LPS is another really important one for you guys in micro. Lipopolysaccharide. So saccharide tells you what right there is, okay, that's the sugar. We know it's a sugar. It's in the carbohydrates. But there's also the lipo part. So this is a big sugar with a lipid attached to it. So it has some lipid part to it, and it has some carbohydrate part. This one is going to be in the outer membrane of specifically gram-negative bacteria. And you guys are going to do a Gram stain. It's named after Dr. Gram. It makes your cells purple or pink. And these cells won't stain purple or pink because they have this lipid layer. It won't let the stain come in. So when you are working in a laboratory and you're trying to decide what kind of pathogen am I dealing with, then one of the first tests you'll do is say, is it gram-negative or positive, to classify it, and then you'll do more tests from there. So you'll have fun with that. So LPS is good for the bacteria. It's protection and it's support. It kills us, literally kills us. So what happens is someone goes to the doctor for a bacterial infection. It's a gram-negative bacteria. They get a bunch of antibiotics. Great. It kills all the bacteria very rapidly. What ends up happening is LPS releases into the bloodstream in high volume, and it causes a major immune reaction throughout the entire body. It's systemic. What your body does is it releases this. You don't need to know this, but you guys are scientists, so this is good for you to know. It's called tumor necrosis factor alpha, this thing that dilates your vessels, okay, and helps your white blood cells come in and attack. But that's released throughout your entire body at one time. So every vessel in your body goes, woo. What do you think is going to happen to your blood pressure? Down it goes. You have no blood pressure, you have no blood pumping, and you go into shock and you die very quickly. So that's septic shock. So that's from LPS. So you got to be careful with these, and you got to take your antibiotics the right way. Okay. You're like, I'm going to read those labels. Those was all of our structures. Okay. These are the ones that are energy, that are, we know glucose is a good energy source. It goes right into the cell, but what about the polysaccharide energy sources? There's two types, one for plants and one for animals. The plant one is starch. So this is your potato, your sweet potato, your carrot, anything you're eating that has starch in it, that is a great energy source for us. We eat that all the time, right? That is when somebody says load up on your carbs before a big race. That's what they're telling you to do, is eat a bunch of starch. So plants use this for their energy source, but we can too. Glycogen, the plant, by the way, stores this starch for its energy source. It doesn't have fats. It uses starch for long-term storage. Starch is really like big and clumpy, so as a plant, you're not running around. This is fine. But for an animal, you don't want starch as your long-term energy storage. That's why we have fats that are more dispersed. But for a quick energy source, your cells are going to take up glucose. So let's say you guys ate your breakfast and you get your glucose into your cells and they're happy. The next thing your body is going to do with your glucose is form glycogen. Okay? So glycogen is very similar to starch. These are both monomer units. The little green circles are glucose. Just glucose, but the way that they're attached to each other, they have a different glycosidic bond. So the structure should actually be different. And this is not a great picture. This is a helix. This one is all branched off. So it looks different when you look at a bigger picture of it. So glycogen has different structure. In your body, you're going to put it in two places. You're going to put it in all your muscle cells because your muscles need a lot of energy. We use glucose to make ATP, right? So your muscles are going to take all this glucose and they're going to store any extra as glycogen in case they need it. Your muscles keep it for them. So even if your brain is starving and has new glucose, your muscles are not going to give up the glycogen. Okay? They don't have the ability, but your liver does. So your liver also has glycogen stores, and it's reading your bloodstream. So your blood goes through the liver, and the liver is going to say, hey, you don't have enough glucose in here, so it'll chop up glycogen and put glucose into the bloodstream to feed your body. So thanks, liver. So that's our quick energy source. If we have glycogen reserves and we have glucose, then we're going to take extra glucose and make it into fats. This one I kind of stuck in the back because it's not specifically for structure and it's not really for energy, but it's part of bacteria. So we're going to talk about it, the glycocalyx. So this essentially means a sugar coating, and it's on the outside of the cell wall. So the bacteria has a plasma membrane, it has a cell wall, and then it has this sugar layer. It helps protect them from your white blood cells, okay, your phagocytes. It can help them attach onto your cells. It can help them attach to each other. It can help them form a biofilm so they stick. And there's two different ways they can form this glycocalyx. They can form a slime layer, which is just kind of diffuse and sort of stuck on there. You know, it doesn't look very organized. Or they can form a very distinct gelatinous layer, so it covers everything, and that's called a capsule. So we'll get into that in later chapters. So that's carbs. That's one. How you guys feeling? You guys look good for 8 a.m. I didn't know how this was going to work out. You're not looking bad. And lipids are weird. These are not macromolecules. So what do they not have? They don't have a monomer unit. See, I wrote down here, no monomer unit, no repeating. There's no monosaccharide. And you notice with almost all of those that we talked about, it's always glucose. It's just the way you stick the glucose together can change the structure. These are still big. Lipids are huge, okay? But they're just not a repeating unit. They still have a dehydration reaction. Oh, I'm going to get a workout in this class. To build their structure, we're going to stick things together. So the specific dehydration is called an ester linkage. And you can say bond. You can say an ester bond, or you can say glycosidic bond. Just more commonly, we hear linkage. Lipids are weird, okay? Because here I am telling you about carbohydrates, and I said you have simple and you have complex. They're all carbohydrates. But lipids is the broad class, but there are 3 different types within that you're going to learn. There's actually 4. There should be, we forgot steroids. Steroids. Man, formatting. Come on. Wow. Let me see my problems. Okay, so steroids. So there's 4. So they're all lipids. So you cannot use, students like to use the word fat and lipid interchangeably, and that is not correct. A fat is a type of lipid. So is a wax. Okay? So the candle that you're burning is also a lipid. So you have to say that these are types of lipids. We're going to start with fats first. So first, a little love for the fats out there. They're good for you. Okay? And this is why I'm always yelling at students to eat and to eat balanced, because if you don't want to eat fats, that's great, but they make up your cell membrane. They make up a lot of the membranes that go around your neurons. Okay, so again, if you want to be smart, you should eat. So your brain has glucose, and it has really quick electrical conductivity, because if you don't eat right, your body doesn't have these building blocks to make what you need to function. Okay, so fat-free diets are bad. You should never cut out something Mother Nature created. That's a bad idea. She knows way more than we do. Okay, it's like taking out organs we don't know. Like, bad idea. Okay. So another word for fat, and you can use these interchangeably, is a triglyceride. Okay, what does tri mean? You guys know that? 3. Okay, so that word's important, and glyceride. You're going to see that is for glycerol. So here's the structure that, yes, you need to know the structure. Glycerol is in this gray box. It's an OL, so guess what this is? An alcohol. Remember we were saying if you had hydroxyl groups that we called you an alcohol? There's actually an alcohol group here, here, and here before it connects. Okay, so before it's going to attach to anything, there's 3 hydroxyl groups on there. So glycerol is polar. It can mix into water and dissolve. The other component, the reason we have the number 3, is for something called a fatty acid. Be so careful. These are when I tell you words mean things. A fat is not the same thing as a fatty acid. Okay, fatty acids make up the fat, 3 of them. So in the yellow, this is 1 fatty acid, 2 fatty acid, 3 fatty acids. So if I ask you to make me a triglyceride, you're going to say, no problem. I need 1 glycerol, and I need 3 fatty acids, and I'm going to stick them together doing these ester bonds. 1 here, 1 here, and 1 here. Okay, so 3 ester linkages to make one fat molecule. What is the function of our fats? For us, long-term energy storage. For other animals that live in the Arctic, it might be insulation. We're kind of coming out of winter, so we can also say insulation, because this has been cold. So if you put on some pounds over Christmas, you can be like, well, that was my insulation. Okay. And then the important thing I was telling you, these are part of your cell, well, these ones wrap neurons. Okay, so that's good. But I want to show you again how you make this. So here's glycerol. You do not need to know that it's a 3-carbon alcohol. You just need to know if I said, what is a fat molecule? You could tell me it's glycerol and 3 fatty acids. But I want to show you these functional groups. Okay, so here's the hydroxyl group. You're going to lose that hydrogen. This is called a fatty acid. Acid is key because it's telling you a functional group. In red, I've highlighted the functional group that behaves as an acid, the carboxyl group. Remember, it has a carbonyl and a hydroxyl. Okay, so it behaves as an acid. That's where it gets the name fatty acid. Why do we call it a fatty acid? Because this is the fatty part, a hydrocarbon. This whole section is nonpolar. Okay, it doesn't interact with water because remember, carbon and hydrogen have equal electronegativities. This is polar. This is all polar. Okay, this is an amphipathic molecule because it has sections of polarity and nonpolarity. So here's where you're going to lose, and it doesn't matter who's given a hydrogen and who's given a hydroxyl group. So you're going to lose a water. That's your dehydration reaction, and you're going to form a bond between this oxygen and carbon. So it looks like this. So let's talk about these fatty acids. They're going to define your fat molecule. Okay, so you've heard of a polysaturated fat or monosaturated or unsaturated fat molecule. You're actually talking about the fatty acid. Okay, so these can be anywhere from 16 to 18 carbons in length. Okay, they can be really long. Okay, and this is just repeat, so you guys already have that on there. So we're going to have a hydroxyl and a carboxyl group attached for an ester linkage. These fatty acids don't have to be the same. Okay, so the fat molecule can have different fatty acids. These ones are the same, but this one could be longer, this one could be shorter. We could put a double bond right here between these 2 carbons. Okay, so you can change those fatty acids in the fat molecule. So these acids, we already said they vary in their length, the number of carbons. They also can change where they have a carbon-to-carbon bond. An unsaturated fatty acid has no double bonds in these carbons in the hydrocarbon. Okay, in the hydrocarbon. Students always say, well, there's a double bond right there with that carbon. Well, that's the oxygen, number 1. Number 2, that's the carboxyl group. So when I am saying carbon-carbon double bond, I mean in the fatty hydrocarbon section. So see how these are all single bonds all the way across? That's a saturated fat. Did I call it unsaturated? It's saturated. Okay. What is it saturated with? Hydrogen. Okay, because instead of being bonded to another carbon, it has a spot to put a hydrogen. Carbon's valence is 4. It can form 4 bonds. Okay, so here, anywhere it's not bound to a carbon, it can put a hydrogen. So it has as many hydrogens on it as it possibly can. An unsaturated fatty acid is classified that way as long as it has 1 carbon-carbon double bond. Okay. So it's not as saturated, right? See how it's going to have less hydrogens than this one? Because these 2 carbons are sharing a bond. There's no hydrogen here or here. Does that make sense? It can have more than 1. Okay, this is a mono unsaturated. It could be poly unsaturated. So that's going to define the fat molecule. So here we're looking at a fat molecule, and this is unsaturated fatty acid, unsaturated Saturated, sorry. Help me out. It's early. This is your unsaturated. Okay, so just one carbon-carbon double bond has changed this fat molecule into be a mono unsaturated fat. Okay, if it had more than one, it doesn't matter where they're at. It could be in here, it could be here, here, here. As long as there's more than one of these, it's called a polyunsaturated. Okay, if I went back here, this is a saturated fat. Does that make sense? And here's just some more pictures. So you can call it a fat molecule, you can call it a triglyceride, and then this is showing you some different pictures. Notice this, how this kinks up the tail if there's a carbon-carbon double bond. Carbon-carbon double bonds, because the carbons are closer together, are much more rigid. So that forms that kink in the tail. Okay? And that changes the complete structure and how it functions in your body too. If you have an unsaturated fat, there's more space between them. So this is one molecule. If you had a bunch of these and they're sticking out like this and it's very rigid, they can't get very close together. So think of olive oil. Okay, so the fat molecules are in there, but they're not very dense because they can't get any closer together. So at room temperature, even though it's room temperatures and there's not a lot of energy in the air, they are going to stay fluid. But if you have something like butter, saturated fat, because there's no kink in that tail, they can pack in very, very tight. So if you just think of, them all squeezed in, okay, they become much denser because they lose their energy, they slow down, they all become dense, and they form solids at room temperature. Okay, so that's your fat. The second one is a phospholipid. This is making up your cell membranes. There are lots of different types of phospholipids. We're just giving you the generic. It's 2 fatty acids instead of 3 and a glycerol. It's a little bit, and of course a phosphate group. Man, I'm like not on it today, huh? And a phosphate group. So 2 fatty acids, a glycerol, and a phosphate. It's a little bit, like I was saying, a little bit more complex than that, but that's the basics. That's all you need to know. So you should know the difference between this and a fat molecule, which is 3 fatty acids in 1 glycerol. This is also an amphipathic molecule because this is all up here, polar, 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 and this is the hydrocarbon, which is nonpolar. We say the polar group up here, we call it the polar head because it's shaped kind of round. We call it a charged head. Right? It's charged because it has these negative charges on it. So here's your charged head, and then here's your nonpolar tails. If you drop these into water, let's just say you drop a couple, okay, part of it is going to interact with water because it's charged, and it likes the charges on the water. But the little tails are not. There's no charge. So they get repelled away. Okay, so generally what they'll form is this structure called a micelle. So you'll see the little heads facing out and the tails facing in. Okay, and they can form a bunch of different things, but in our cell membranes, the way that they work is they form a bilayer. It's called a phospholipid bilayer. So 2 layers of phospholipids. Inside of your cell is water. Cytosol is water. Outside of your cell, on every single cell, is water. So everything dissolves into water, has to cross this membrane, and then dissolve back into the water. So the way you have structured, okay, to give your cell a structure is to have it where the polar heads on the bilayers face out to the water. There's negative charges here. Okay, so it's going to be attracted to the hydrogen. And then you have this layer in the center that's nonpolar. Okay. The characteristics of these phospholipids, so do they have an unsaturated or saturated fatty acid tail, are going to affect the fluidity of the membrane. These move around. Like salad dressing move around. That's how much these little phospholipids are moving. Okay, so that helps things get in and out of your cells. So if they're not moving at all, let's say it's all saturated fats, fatty acids, and they all pack in tight, and it's cold, and you're a cold-blooded organism, and they all pack in, these are going to get very dense, and nothing can get in and out, and that organism will die. Okay. The other thing, and that's affecting your permeability, what can cross. Okay, if it's too hot, it's going to be too fluid, then anything can get in and out. Okay, you lose your structure. This is what it looks like. Okay, and this would be showing you one that has more fluidity because it has space from these carbon-carbon double bonds. This one would be viscous, like syrup. Okay, it doesn't move enough. Okay, so you're going to lose your selective permeability. Steroids we'll get through quick. This is what a steroid looks like. It's 4 rings, and then it has your functional groups hanging up. The most important one is cholesterol. Cholesterol is one of these things that gets a bad rap. There's 7 different types of cholesterol, and there's only one that's bad. So cholesterol, one of the important things it does is it inserts into cell membranes like you're seeing here, and it helps keep them fluid, especially in an animal. Okay, so this is good for animal cells. Ergosterol is important in the cell membrane of a fungi. Okay, it can be important in cell wall deficient bacteria. In your bodies, it's important because it's a precursor to a ton of hormones, like testosterone and estrogen, and all of the prostaglandins. Okay, lipids, the last one, wax. Okay, we don't have wax, but a plant cell on the outer leaf, do you ever see the shiny part of a leaf? That's waxy. Okay, and a lot of animals like a duck. Have you seen the ducks swimming around in the water and like it's freezing? How are you hanging out in there? Because it's not on their skin. They have wax on the feathers that's helping it go off. This is an ester that forms between an alcohol and a fatty acid. Okay, you can warm it up. It's going to be soft, and then it's going to be waterproof when it's cold. Okay, and we see them in bacteria. They can cause pathogenicity in people. So it's just a quick little thing on waxes. All right, proteins. Okay, the monomer unit for this is back to we're having a repeating unit, is an amino acid. So there's 2 words there, amino and acid. That's your functional groups, an amino group and a carboxyl group. Okay, so when you look at an amino acid, you should see these 2 functional groups. I shorthand amino acids with 2 capital A's. Okay, so that's not American Airlines, although they are my favorite. That's amino acids. There's 20 basic amino acids that exist in nature. There's some other weird ones out there, but 20 basic. You don't have to memorize them. I'm just letting you know that. They stick together in different order, okay, and in different amounts to make millions of different proteins. So think of it like 26 letters of the alphabet. You don't use all 26 letters in a word, right? You can use 3, you can use 10, you can use 2 L's or one O. It's the same for amino acids. Okay, so it's their order that's important. When they link together, like those pop beads, the polymer is called a polypeptide. If you have just a couple amino acids, it's called a peptide, like maybe 8 to 12. Okay, don't memorize that number. It's just a couple of them. You'll see that term is sometimes in a scientific journal or in a protocol. It'll say a peptide. It's just a couple amino acids. Polypeptides are referred to as a protein. So you could say polypeptide, you could say protein. This is your basic structure of an amino acid. So all 20 have this. They have what's called an asymmetric carbon. That is a carbon that has 4 different groups attached to it. Okay, glycine is technically doesn't have this, but it's okay. You don't need to know that. Okay, so you have 4 different groups here. It's always a carbon. You're going to have 2 functional groups, the amino group. Do you remember this? The nitrogen that acts as base. And then the carboxyl group that acts as the acid. So this gives amino acids their name, the amino acid. Okay, they have a hydrogen, and then across from the hydrogen, you're going to have this R group. The R group, or the side group, is what defines the amino acid. Okay, just like I said, how do we define an element? How many protons it has, says it's carbon or hydrogen or nitrogen. The R group is what calls this glycine or lysine or proline. Okay, that's how we're going to name this amino acid, because you all have these sections. What makes you different? This one group. Okay, so these are your 5 parts. The carbon, the hydrogen, the amino group, carboxyl group, and then this side group that defines the amino acid. This part that we're talking about is called the backbone of the amino acid. So we're going to talk about protein folding, and it's going to say hydrogen bonds between the backbone. You're like, what the heck is that? Everything but the side group. There's 3 classes: nonpolar, polar, and electrically charged. Okay. We're basing it on the side group. So a lot of times students will look at an amino acid and say, well, how can this be nonpolar? This is polar, this is polar. That doesn't make sense. You have to remind them you're only looking at the side group. This is what defines it as nonpolar. These are methyl groups. Remember, carbon and hydrogen have equal electronegativities. Hopefully, if you got anything out of this lecture, you know that, right? I've said about 15 times. Okay, so these are all nonpolar groups. No, you don't have to memorize this. These are your polar amino acids. So you're going to see hydroxyl groups, sulfhydryl, amino, carbonyls. Okay, so those are all in there. The last one are electrically charged. These have either carboxyl or amino groups that are ionized. Okay, so you're going to see them with an extra electron or an extra proton. So we start sticking these amino acids together. We're going to form a polypeptide. We're going to do it through a peptide bond. Okay, so there's your dehydration reaction to form the covalent bond. So here's one amino acid, here's the second. You should know how this forms. It very specifically forms between the carboxyl functional group and the amino group. Okay, you're going to lose the water. You don't have to draw the structure out. Just know which 2 groups are interacting. It's the carboxyl group and it's the amino group. Okay, so that's your peptide bond between carbon and nitrogen. Every protein has a different linear sequence, meaning a different order of those amino acids. There's 4 ways that we're going to fold up our protein. Okay, there's 4 levels of this structure. Okay, and of course, you get to learn them all. They all have primary, secondary, and tertiary structure. There are some really big proteins that have quaternary structure. So we're going to go through each one. Primary structure is just the sequence. Okay, so what amino acids in which order? Okay, so we said that's just like the letters in a word. Okay, this is determined by your DNA. Your DNA codes for which amino acids to put in which order to make the protein. When you look at it, it really does look like pop beads on the string. Okay, and on one end, you're going to have the amino end sticking off, and on the other, the carboxyl. That's because if you go back to this picture, you can see it. Here's the amino end. If I attached on another one, there would be a carboxyl hanging off. And then another one, a carboxyl hanging off. So when you have, let's say, 100 of these, you're always going to have the amino end and the carboxyl end. Okay, and that gets important if you ever take cell phys of how you put a protein into a membrane or wherever you're going to put it. Okay, so the bond is this peptide bond, and it is between your amino acids. That's the easy one. Secondary structure. Okay, this is specifically hydrogen bonds. That's what H bond is. Okay, so these are not covalent. These are hydrogen bonds, and it's specifically between the backbone. Do you guys remember what the backbone is? It's everything but the side group. And it forms these 2 unique structures, beta-pleated sheets and alpha helix. Be careful, this is not a double-stranded DNA helix, it's a single helix. Okay. Not all proteins have both of these. They usually have one or the other. Some can have both. Okay, so you're going to see these structures, these helixes, and this almost looks like that crinkle paper that you get in a gift wrapping. The third level, okay, this is the side groups interacting, these R groups, which remember is unique to each amino acid. So within the huge, long polypeptide, you get all sorts of bonds, every bond. You can have hydrogen bonds. You could have covalent bonds. Here's a disulfide bridge between 2 thiol groups in these amino acids. Here's an ionic bond. Okay, here's something called a hydrophobic interaction. These are all methyl groups. They don't interact with water. So if you were in the bloodstream, these might kind of fold in away from the blood, just like we saw with the phospholipids. Here's a hydrogen bond. Okay, so these are all the side group interactions. This starts to give the protein its actual shape, which gives us the function. So here you can see an alpha helix, you can see beta-pleated sheets, and then you see the big overall structure of the protein. For the really big ones, they're comprised of more than one polypeptide. That means you're going to take the big, long string, and you're going to make it all and fold it up, and then you're going to take another big, long string, and then you're going to stick them together. Collagen, I know you've heard of that before, has 3 polypeptides. So here's 1, there's 2, there's 3, and then they weave together. The whole structure is considered one collagen protein. Okay, so pieces of it are just pieces of it. Hemoglobin is another really important one for you guys. It has 4 different polypeptides, 2 beta chains and 2 alpha chains. Okay, so all 4 units together make one big protein molecule. So proteins are really cool. They're very complex. Here is something I made for you. Okay, it's the structure of proteins. Okay, the levels, primary, secondary, tertiary, quaternary. What kind of bond is there? This is covalent, hydrogen. These are all types of bonds. And who is it between? This is amino acids. This is the backbone. These are the side groups. And this is polypeptides. Isn't that a lot easier to study it that way? Yes. So that's a good hint for you for studying. Make a chart. A quick note on enzymes. We're going to talk about enzymes. We'll say, oh, well, that's it. It has an enzymatic function. They're proteins. They're made out of amino acid. There are these really weird enzymes that are made out of RNA. They're called ribozymes. They exist. I'm letting you know they exist. But 99% of all the other enzymes are made out of amino acids. They're proteins. So you can have a protein like collagen or hemoglobin. They're not enzymes. But if you hear the word enzyme, think, well, that's a protein. Okay. Their job is to speed up a chemical reaction. So let's say you eat some sugar. Right? It's going to go in your stomach, and enzyme sucrase is going to attach to it, wiggle and jiggle it, and pull it into glucose and fructose, break it down. That same reaction could occur if you dump sugar on the sidewalk. Without the enzyme, it's just going to take a whole lot longer. Okay, so they just speed up reactions that were going to occur anyway. The native state, this is something you might hear. Okay, that's when it's functional. It's formed and it's all folded up the way it's supposed to be. Okay, so if you have the native state of a protein or an enzyme, it's functional. Okay, oftentimes, we as humans make a lot of errors in our laboratories because we denature our proteins. That means we start to unravel them. Normally, when we're denaturing, I'm going to go back and show you, we're affecting these levels, quaternary, tertiary, and secondary. Okay, so we're just kind of unraveling the structure enough that it's not functional anymore because its function is to attach specifically by shapes, right, into something. So if we unfold it, we've ruined it. And a lot of times how we unfold it is we hold it in our hand. When I was in grad school, there was a girl who would walk down the hall with enzyme for her molecular work in her hands. So she would go get it out of the -80 freezer, -80. It's cold. It's supposed to be cold. She put it on ice immediately because it's an enzyme. Nope, she would hand it and walk down the hall with it. She did not graduate. We'd always be like, you should probably use ice. I don't know. She's like, my experiments aren't working. Yeah, because you're denaturing every enzyme you touch. Okay, so if you work in a lab, put everything on ice. Here's what an enzyme might look like. Okay, it doesn't change its shape. You'll hear that term, it's not consumed. That means it doesn't do anything. It just attaches to something. We call it the substrate. Attaches to the substrate, helps break a bond, for example, releases it, and then picks up another substrate and just keeps going around and around and around. All right, so that's it. We're getting there, man. We have one more, nucleic acids. This is the easiest one because you've already heard of DNA and you've heard of RNA. Okay. Make sure you know what these words stand for. I was once appalled when a senior told me he had no idea what DNA was. He's like, I know what it is, but I don't know what the letters mean. It's deoxyribonucleic acid, or ribonucleic acid. DNA is able to direct its own replication. So it replicates itself, which is super cool. It directs the synthesis of messenger RNA, okay, which then makes proteins. However, the more we learn, the more we know we don't know. Right? So now that we've sequenced the human genome, we found out that DNA all of it, right? We have all this DNA. Only 2% of it is actually translated into a protein. So what the heck is the rest of the DNA doing? It's making RNA. RNA is doing everything else. So RNA is actually what's in control. We don't actually know everything yet. So you guys have a job to do. You have to go out there and do research on RNA. Okay, so structure is important. Okay, your repeating unit is a nucleotide. Okay, for both RNA and DNA, we call it a nucleotide. That's your monomer. So you're going to attach this nucleotide over and over and over. You know what I'm thinking? The bond is a phosphodiester bond. I should have put that on there for you. I'll reload this right after class so that you guys have it updated. I'll make sure I put that in there, that your bond that you're going to use, your dehydration reaction, is a phosphodiester bond. That's what attaches these nucleotides. Okay, a nucleotide is made of 3 parts, and you should definitely know this. There's a nitrogen base. Okay, we call it a base. That's just a chemistry term. It's a ring structure with nitrogen in it. There's your ribose, so your pentose sugar. Okay, there's ribose. And then a phosphate group, your functional group. So phosphate, ribose, and this nitrogen base. The polymer, if you attach a bunch of them, is a polynucleotide. Okay. DNA, if you say just DNA, you're implying there's 2 strands. So here's one polynucleotide, here's the second polynucleotide. In between them, remember the dashed lines? Hydrogen bonds. So a polynucleotide has all covalent. In between the 2 strands, hydrogen. RNA is single-stranded. Okay? So this has, my gosh, wow. I can tell that I was just in a hurry doing this. One polynucleotide. Does that make more sense? Like, damn it. Sorry. You guys will be so happy when Dr. Jackson comes and she already has this all done for you. Okay, the sugar, let's talk about the sugar first. Okay, the ribose. It's ribose in both DNA and RNA. It's just a little bit different. This is normal ribose. Okay, so here's your carbons that aren't listed. 1, 2, 3, 4, and this is number 5. So it's the second prime carbon has this hydroxyl group. In DNA, it's missing an oxygen. So deoxygenated, without oxygen, which is where the term deoxyribose comes from. Okay, so now you know. So if I give you 2 DNA nucleotides or a nucleotide and I ask you, is this DNA or RNA, that's one of the ways you can tell. Say, oh, well, there's no oxygen down here, so I know that's DNA. Or there is an oxygen, that's got to be RNA. Okay, what about this nitrogen base? This is what the bases look like. There's 4 of them. This is what defines the nucleotide. They all have a phosphate, and they're all going to have either ribose in RNA or deoxyribose in DNA. So what makes one nucleotide different from the other is this base. Okay, they can be a purine type of base, which means they have a 2-ring structure. Okay, that's adenine or guanine. Or they could be a pyrimidine. And I tried to put a little pyrimidine so you can remember how you say this. Pi, because it's going to help you. It's just 1 ring, like a pie. Okay? Okay, the specific bases are thymine, cytosine, and uracil. So an easy way to remember, how do I know who are the purines and who are the pyrimidines? If you like to eat pie, you have to cut the pie. Okay, so CUT, that's these letters. Cytosine, uracil, thymine. That's a really easy way to remember. Those are pyrimidines because you're going to cut the pie. This is what I'm seeing right now. So either you're just dead or you're like, that's stupid. You'll thank me on your test. So of these, whether you're DNA or you're going to be RNA, you're going to have cytosine, you're going to have guanine, you're going to have adenine. The second difference between these types of nucleic acids is that RNA has uracil, DNA has thymine. Okay? DNA is a more stable structure than RNA. Okay? RNA degrades faster, so it has to do with The ribose and the uracil structure being a little bit different. And that's just functional. You don't want it hanging out in your cytoplasm. You want to get it out there. You want to make your protein, and you want to degrade it quickly. DNA you don't want to degrade. That's your good copy. In DNA, these bases attach to each other. We call it complementary base pairs when they form their hydrogen bonds. Okay, I drew this like this so you can see, but remember, these are not covalent. These are hydrogen. It's always a pyrimidine to a purine. Okay, so it's always one ring to two rings. And you're seeing it here. So thymine always will complementary base pair through two hydrogen bonds to adenine. Cytosine always bonds to guanine with three hydrogen bonds. Okay, so if you have thymine that goes with guanine or cytosine with adenine, you're going to have a mutation. That doesn't work. Okay, so this is what it looks like in the DNA. RNA, what does it do? Your DNA is your genetic code, right? That codes for your proteins, and now we know it codes for RNA that does a lot of other things. The major types of RNA that you're going to learn about, message, that's when you're going to actually copy your gene and then translate it. tRNA, transfer RNA, brings in amino acids when you're making a protein. Okay, so it's the one that's delivering. Okay, this is like Amazon Prime. Okay, brings it in very quick. rRNA is ribosomal RNA. It makes up the ribosome that is holding the message and brings in the transfer RNA and lines everything up. Looks like this. Okay, this big green structure, this big circle, that's the ribosome. This strip is the message. Okay, and then these little guys here, each one of these is a transfer RNA with its amino acid. So the ribosome is holding the message and then lining up the transfer RNAs to put on the amino acids in the right order to make a protein. So DNA, make your message, take it out here, ribosome, make your protein. ATP, we mentioned ATP here. It's adenine triphosphate. Why the heck are we talking about this in nucleic acids? Because it's the same structure as a nucleic acid. It has the nitrogen base, adenine, it has ribose, and it has a phosphate. This right here, if you just attached it, would be an RNA nucleotide. But we're also going to add two more phosphates. So it's triphosphate. This is our energy source that we can use in our bodies. If we take off a phosphate, then we have two phosphate groups, diphosphate. If we take off two, we have one. We would have monophosphate, AMP. Okay, so you're going to hear about ATP a lot.